Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
We have the last talk of this very stimulating day, and the talk is by Vladimir Minin from University of Washington. The title is Simulation-Free Algorithms for Mapping Evolutionary Innovation onto Phylogenies. Thank you.
And thank you for the invitation. So my microphone is working, right? Okay. So it's being the last speaker has some disadvantages, advantages. Disadvantages, you lose some audience. The advantages are now I can say, well, remember what Joe Felsenstein told you 2 hours ago, and build upon that. As a matter of fact, in this talk, I will continue the this third part of Joe's talk when he said why we need, what happens when we infer phylogenies. What do we use them for? This is usually for some of the evolutionary biology labs. This could be the last step in the analysis, but most of the time you really want to do something to do more. So throughout the talk, I will assume this standard model that we've seen before. It will be finite site model. So Yoon talked about infinite site model, and several other people mentioned that before too. So we will assume a possibility of more than one mutation on each branch. And the model is a standard continuous time Markov chain model parameterized by some rate matrix. So here I just present a particular parameterization. Doesn't really matter which one you use. This is a particular popular parameterization. The probabilities of mutations on a particular branch are calculated by inserting those times or branch length into the transition probability matrix, which you obtain by exponentiating your rate matrix multiplied by t. And we again assume IID distribution of columns in the alignment of sites. Again, you can relax it a little bit, but not too much. And this will be our object, a phylogenetic tree. Again, we observe data on the tips. So those internal nodes of tree are hidden. We don't know which states they are. So for example, one question that people ask when they reconstruct phylogenies, which I'm not going to talk about too much, is can you reconstruct the ancestral sequence? And there's interesting work done by several labs which reconstruct ancestral sequence and actually try to put this new organism together, or at least new protein together, not new organism. New organism is very, there is one work like that, but mostly new proteins, ancestral proteins, and try to say, well, how different the function was, why it was the way it was, and why it evolved into what we see now. So that's one type of sort of going one step further when you have phylogenies, because without them you cannot really do this ancestral reconstruction. We want to go even further. We want to not only reconstruct ancestral states, we actually want the full history, right? That would be nice. If we knew exactly where a mutation happened on the tree, at each time point, which kind of mutation. So for example, here, with like a T mutated to G, and then nothing happened here, nothing happened here. So this data pattern can be explained by just one mutation. And it would be nice if we could place this mutation exactly somewhere in the tree. So, but as Joe Felsenstein mentioned, so here's my first remark. Remember what Joe said? The parsimony method can do it pretty easily with this minimum evolution principle. If you try to find a tree that minimizes the number of mutations that you observe, you can sort of infer which kind of mutations and where they occurred if you assume that there are a minimum number of them. Unfortunately, when you deal with in this model-based framework, this is not the only pattern, right? I can also have I could have had here a G which mutated to T and nothing happened here, and so that would be perfectly fine. So the probability, let's say the posterior probability of that mutational history would be probably equal under these conditions. So we don't want to discard that. We want to somehow summarize the distribution of all those mutational histories. So we want to look at the full picture. Well, it's pretty difficult just from the bookkeeping point of view and from other point of views to summarize the full history. Usually we don't really want the full history. We want some summary of the full history. So that will be the full history. If I knew that T was all the way up here, then changed to G, and then G was all the way here and all the way here. So you can color your trees by state, for example. But summary will tell you the number of certain mutations that occurred along the tree. For example, I ask you how many times T mutated to G. This is not under in model-based framework. If I give you model parameters in the tree, this number is not fixed. This is a random number. It has a distribution. So we would like to characterize this random number by moments or probabilities, generating functions, you name it. The same with you can put some rewards. You can reward your state, your trait for being in a particular state. For example, the simplest reward would be I just want to count how much time on a tree our trait spent in state T. What is this time? And then I could compare I can divide it by the total sum of branch length, which is called tree length, and that would give me a fraction of time a trait spends in a particular state. So we want all of those things, and right now and in this talk, objective will be just to take this expectation. So you have your summary, which could be either this or this, and you want the expectation conditional on your data. So currently, if you want to use model-based approach, the only available method is simulation. So there's a software called SimMap, and the method is developed by Rasmus Nielsen in 2002. We would like to avoid simulations. Well, it turns out that this problem is, from a probabilistic point of view, is simple enough that no simulations are needed at all. So let's consider this first summary that I mentioned, the counting process summary. So how it's defined. So assume now just a simple one-branch calculation. So you forget about phylogenetic tree, just concentrate on one branch. Suppose I give you the conditions that you must start at 1, you must end up at 1 at the end of the branch length, of the end of your time interval t. And I want the expected number of a certain kind of labeled mutations. So for example, in this simple 3-state continuous time Markov chain, I want to label only mutations 1 to 3 and 2 to 1. That's it. So those mutations define a counting process on the real line right here. And I want to to characterize this counting process. So in this case, in this particular case, this set of labeled mutations will consist only of 2 pairs. So this is ordered set. I don't want to count any 3 to 1, only 1 to 3. Your rate matrix. Remember, lambda is your rate matrix, your regular rate matrix that you have when you deal with continuous time Markov chains. So basically, what happens? You just strip everything from this Markov chain except those rates that you want to label. So here we are left only with lambda 1, 3 and lambda 2, 1. Everything else is 0. So this turns out to be an important matrix in the calculations. And then you want to compute this expectation, which is a little odd in this formulation. What you really probably want intuitively is expected number of those counts conditional on starting and ending state. But clearly those two expectations are related. You just divide this expectation by the probability of I going to j, and you get back the full conditional. So there's no real problem with having expectation written in this joint form rather than conditional form. Then you put them all together, stack in the big matrix E. So E has the same dimension as the original state, in this case 3 by 3. And it turns out that this matrix is simply this integral where lambda is your original rate matrix, lambda sub, so this labeling set, so this auxiliary lambda, times e lambda t minus z. So this is a convolution, simple convolution integral, which I will not go into details how to compute, but it's very easy to compute. There are several methods. Engineers computed by uniformization method and just a definition of matrix exponential. So you can imagine you just stick the definition of matrix exponential here as an infinite series, and you just truncate those series, and you get some approximation back. In fact, in evolutionary applications, it's even easier because most of the time we assume that those models are reversible, and for reversible models, you can just compute the eigenvalues and eigenvectors and get the exact matrix E. No approximations are made, except you may, in some cases, you will need to approximate your eigenvalues and eigenvectors, but that's fine. That's done anyway when you compute probability and likelihood. Well, that's first process, right? So the counting process. So I hope I convinced you that you can do one branch calculations for counting. Count all possible transitions that you want, labeled transitions of a Markov chain. Now the reward process, it works kind of similarly. It turns out, well, it turns out actually that for fully observable Markov chain, both counting and sort of the reward process form the minimal sufficient statistics. So there's some deep connection with the with probability statistics here, I think. Well, suppose your weights are simple. So your weights are is a vector 1, 1, 0. One weight for each state. So in this case, it means that you just count, you just keep track of how much time you spend in state 1 and 3, and you lump them together, and you get your evolutionary reward in this case. So you just completely ignore state 3. And you can put any real numbers here. I will, in most applications that I will talk about, you will, well, actually in all of them, we will use only zeros and ones. So in this case, the auxiliary matrix is even simpler, and the problem actually is a little simpler than the previous one. It's always diagonal matrix, and you put weights on the diagonal. So in this case, we just put ones on the diagonal. And it turns out the integral looks exactly the same way. It's exactly, only the auxiliary matrix changed, but the shape, it's the same convolution. Exactly the same methods apply. If you have eigenstructure of lambda, it's again computable without any approximations whatsoever. So it can be done very quickly. So that's nice. Now, we need to project those calculations and propagate those local calculations globally in a tree. So I will need some notation here. So I will be a set of internal branches. E will be a set of terminal branches. If I have a branch B, I will denote the parent of branch B by P of B and child by C of B. We will have an arbitrary. We'll put a root somewhere arbitrarily in some sense, but and that will define the direction. So parent and children will be well defined. So if you look at the complete data likelihood, so you have D is the observed data, vector I is those internal nodes that you never see. Then you would simply multiply all possible transition probabilities, and that's your complete likelihood. Well, if you want to calculate observed likelihood, you sum over those states, and Joe Felsenstein mentioned that can be done via dynamic programming, so that could be done very efficiently. And that's the missing data, and I didn't have a heart to say likelihood here, so I put prior. That's our prior for the internal nodes, which is just a reduction. You just cross over all these transition probabilities that have a D in them. So this notation we have, so this is indexed. This is some particular branch B, parent, child. So you just compute transition probability from parent to a child. So that's it. And this separation to internal and external branches simply to indicate this. We have D's only for external branches. So given that notation, our objective is to find this function, right? So remember that we wanted to find a function of the expectation of this functional summary of our evolutionary history. Well, we consider a particular kind one. We have this additive functional. So I want H to be a sum of small h's, where each small h acts only on single branch. So that actually turns out to be important for our calculations. Then suppose you choose arbitrary branch B, because now I just need to compute those single expectations, right? Expectation is luckily a linear operator, so I just can do single branch-specific expectations, and then just add them up, and that will give me the global expectation. Now, so we reduced our problem just to computing expectation over one branch, but notice we condition the full data, not on starting and ending state. So it's a different problem that I presented before, where I said I give you the starting state, the ending state, give me the expectation. Again, we do this we apply the definition of conditional expectation to arrive at this, and it turns out that this number is easy to compute without notation, or at least present without notation. Both numbers are easy to compute. That's your likelihood that you compute anyway when you do statistical inference. So this part comes for free. So then what you do, it's possible to show that this expectation it looks like this. So we have this ugly formula here. If you define the probability of data, remember I told you the probability of data is this. Here I just simply separated this specific probability, transition probability for branch B star, and I just visually separated it. It's the same statement as before. You can lump it into here and remove this condition B is not equal to B star. But I did it on purpose because, as you see, the only difference between likelihood calculation and this expectation is you replace this transition probability for branch B star with this expectation, which we know how to compute. That's it. Done. So if you can compute likelihood, you can compute this expectation just by using the different number at the particular branch. And we know this number. Well, almost done. So this proves that this is feasible, and you don't need any simulations to do any of that. Notice both h's will do, both the counting and the reward process. So it doesn't really matter which h you stick here. It only matters that we know for those two particular cases, it's very easy to calculate those things. But if you can do those local calculations for more complicated h's, that's fine too. So some consequences of this analogy with phylogenetic likelihood. First, similarly to the invariance of the root placement in the likelihood calculation, we get the same invariance principle. That's what Joe Felsenstein calls a pulley principle. So you can move root around. If you have a reversible model, that's the condition. Then you can move root around, and your likelihood doesn't change. So you cannot really infer rooted trees. You can only infer unrooted trees. It turns out we have the same condition. If I give you a tree and I start changing roots, I will not change the expectations unless this is non-symmetric. So lambda, this auxiliary matrix lambda for the counting processes must be symmetric, because otherwise I break symmetries and bad things happen, and the root placement will matter. So that's something that I don't think have been shown or noticed before. So there's a potential danger. Right now we have this flexible software that allows you to count everything you want to count. But we should realize that the root placement will matter in some situations. Although most natural applications involve symmetric auxiliary matrices. Now, prior distribution, which can be surprisingly useful in many applications, which is just the expectation without conditioning and data. It turns out, that's trivial to compute. So you don't need any fancy tree traversals or anything like that, likelihood calculations. You just have a simple analytic formulas for your parameters. So this, if you're familiar with distance methods, if you plug in auxiliary function, which will be your lambda minus diagonal of lambda, so you just put zeros on the diagonal. So this will be exactly what's called phylogenetic distance. So, which not surprisingly enough, because here we count expected number of total number of mutations that we observe, and that's what genetic distance is. But this is labeled, so it's a slightly more general notion. Now, computational efficiency. Well, we know that likelihood calculations are computationally efficient. Now, if we do likelihood calculation for each branch, we will multiply it and we'll get one extra n term here. So that's not good. Hopefully we don't have to do that. So we need to cache some computations. Now I want to stress again, why do I want to cache the computations? So I want to be able to traverse the tree once or twice perhaps, and then reuse all of these calculations to calculate the expected number of certain mutations or evolutionary reward or you name it, for all branches without doing any traversals anymore. So I just want to do it once or twice. In this case, I will do it twice. And then you will just stick different, you'll just reuse those computations. Because alternatively, you would run your pruning algorithm over and over for each branch and replace the appropriate transition probabilities with appropriate expectations, which is inefficient. So it turns out we can adapt the Schatz-Sinsheimer-Lang algorithm. Not very. I think I'm the first one who uses this name. It's not a very popular algorithm. So Eric came up when he was writing his dissertation. He came up with this algorithm which allowed him to compute partial derivatives of phylogenetic likelihood with respect to branch length, which turns out to be a useful exercise and a useful technique, which is still not used at all in maximum likelihood calculations. But the algorithm is still there. So I realized that what we're doing is exactly the same thing, except the application is totally different, but the caching is the same. So we defined, remember, this audience probably knows very well hidden Markov models. So remember, when you have hidden Markov models, you want to compute the posterior probability of a hidden state. You have this sandwiching formula. When you compute, you calculate your likelihood forward, backward, and then you can glue those partial likelihoods together to obtain the posterior probability of a hidden state, for example. So that's the sandwiching argument. We make exactly the same sandwiching argument here. So what you do, suppose this is our target branch, and we want to calculate for example, rewards here. It doesn't really matter. We can do counting or any other functional. So you break your tree into 4 pieces. So this is your target branch, and those little expectations that we know how to compute, so they're fixed for us, they come here. And then you compute three partial likelihoods. So the standard one, which I call forward, but it's not really clear what's forward, what's backward here. There's no time here, but arbitrarily I call it forward. So it's defined as a likelihood, so you just cut the tree at this particular node, and you compute the likelihood of observing data conditioning on the fact that this is J, this internal state is J. So it will be called, so it's a function of J and the node index. So this particular node will be 4. So this will be F for J. Then the backward likelihood will be right here. It's the likelihood of when you cut the tree like that. It's likelihood of this data pattern, assuming that this tip is real, although in fact it's an ancestral node. But assuming this is a real tip and this is your new phylogenetic tree, and you stick I here. So I is your imaginary data here, not imaginary number. And you get the backward likelihood, which is also depends on which I you stick in. And there is a sort of weird directional forward likelihood. I should get rid of this notation somehow. But so this is likelihood. This basic likelihood. So you just cut the tree in a similar fashion, but also you cut it in half, and you consider only left or right part depending on how you ended up in a tree. So this is the likelihood of observing T given I at node 3. So in this case, this will be simply a transition probability. But in a more general scenario, it will be something more complicated. So then if you pre-computed that, and in order to pre-compute that, it turns out you need only 2 traversals. You need to go first up and then down. So when you go up, you calculate these forward likelihoods, both traditional sort of regular forward likelihood and this directional weird one. And then when you go back, you can calculate these backward likelihoods. I guess that's why the name forward-backward came to my mind. So this is direction forward-backward. So then you can just use the sandwiching formula and sandwich these expectations between those likelihoods and reuse your computations. So you don't have to traverse tree each time for each branch, because number of branches can be very, very, very big. If you have 100 of taxa, you have roughly 200 branches. So you don't want, so 2 versus 200 is a big saving time. So that makes this algorithm especially efficient, which means if you can calculate likelihood for your data, then you can as well do the mapping. John? No, the paper, the Eric dissertation is 98, but I'm afraid I don't. Do you have a reference? I would be interested. Right, and that's what the. Okay, that's exactly what Eric was trying to do, right? So it would be good if they published it too. So. Okay. Right. So the pedigrees we know the reference. That's good. And you can see that this is really nothing, clearly nothing revolutionary. It's a sandwiching formula. If you have a conditional independent structure, it will work for any kind of graph, for example. And well, you probably will have to worry about cycles in pedigrees. Okay, so we can do it efficiently. This Markov chain one branch calculations somehow didn't make it from engineering literature or from pure math literature to any of the evolutionary applications or genetic applications. So up to now, and still we are doing all of these calculations by simulating full realizations of Markov chains and then rejecting ones that do not agree with our data. So we do this extremely inefficient rejection sampling. So just in a summary, so what needs to be done for simulations to work? You need your eigenstructure of your generator. You compute the probability matrix with this generator. You compute expectations with the same generator. So you have to do your eigen. You have to find your eigenstructure only once. Then you reuse it. You traverse tree once to calculate forward backward. Second time to calculate backward. Sorry, forward directional and normal, and then backward. You traverse it second time to calculate backward likelihoods. And then you, at the end, you return this value. So why is it useful? So now it's my second line where remember what Jonathan and David were talking about, detecting correlated evolution. So it's a fairly standard problem in evolutionary literature. You have two traits. You want to know whether they correlate in the evolution. Do you see, or can you test independence? And as Jonathan correctly pointed out, if you do a naive chi-square test, it's not valid. You have this strange dependencies in your traits that you not account for, so you get some spurious results. So this is a very simple example, as simple as it gets. So you have two traits. Let's say they're binary, although the method is not bounded to any binary. Any finite number of states will do. So we have two binary traits. The evolution of each of them is parameterized in terms of this lambda matrix, so this rate matrix. In this case, we have just two parameters. Now, we form a new state space by just ordering them and forming this new state space. It turns out if under the independence hypothesis, if we believe that the two traits evolve independently, or any two Markov chains evolve independently, the new transition probability, the new generator of this continuous time Markov chain can be formed by Kronecker sum or tensor sum of two or more matrices. So if you forget what a Kronecker tensor sum is, that's your definition in terms of Kronecker products. And if you don't remember what Kronecker products are, don't worry about that. It's just a way of making making one matrix out of two in a way that that corresponds to this independent evolution. So, for example, if I mutate 0 to 1, this rate should be B0. It doesn't really matter what this 0, whether it was 0 here in the first position or not. If I do the same with this pair of sequences, I will get the same B0. So the rate of evolution of the second trait is totally independent of what's going on with the first trait. Kronecker representation is simply useful because it's fast to calculate matrix exponentials. If I know the eigenvalues and eigenvector of this, it's very easy to glue all these eigenstructures together, no matter how many you have of those, and you get your eigenvalues and eigenvectors. So it's very quick to do computations with this representation. So we will use these evolutionary reward processes, or in this case, it will be simply dwelling times, as measures of surprise. So we want to design a test, although a Bayesian one, I should admit, which will be an analog of chi-square test. Well, actually, it was designed by John Halzenbeck and co-authors in 2003. We just want to demonstrate that we can compute and do this test without any simulations. So how it works, you try to do something to your regular test of independence. So you define dwelling times of trait 1 and 2, and you define dwelling times of this joint trait, of this trait of a vector of traits 1 and 2. And then you form this expected fraction of time that trait 1 and trait 2 spend in particular state ij, for example, and you consider all possible pairs. So if traits evolve independently, you should be able to just multiply a fraction for trait 1 and the fraction of trait 2 and just multiply them together. However, we can also operate on this product space, and we can also compute those things on this product space without doing these calculations, just directly, right? We have a tree length, and we can compute those expectations, so we can do that. We can do that too, and we call this observed. Notice nothing here is expected, nothing here is observed. Well, you may call this expected, but this is not observed certainly because we're still imputing those histories, so there's no observations here. But it's sort of just by analogy with the test of independence. Then you can form discrepancy measures by taking differences and squaring them, or taking absolute values, adding them up, perhaps divided by expectations, by expected to make it more clearly an analogy. But I was just trying to reproduce something that other people were doing, so we used those discrepancy measures. So in 2003, John Halzenbeck, Rasmus Nielsen. Rasmus was the one who came up with the stochastic mapping and simulations, and thinking actually about mapping those innovations, so mapping evolutionary histories in a model-based framework. And John Bolbach, I believe, was the third author, came up with this test. So we define discrepancy measure by as before, but just by summing those individual discrepancies between expected and observed. Notice what's strange about this discrepancy measure. It's not a standard test statistics. It depends on the model parameters. So we cannot, do the usual frequentist analysis and just, come up with a distribution of this test statistic on the null, then see what we observe and see whether we're surprised by our observation, whether it's too far in the tail of our null distribution. So instead, we do something similar. It's a Bayesian way of doing this. We compute posterior predictive p-values, which say, well, if I suppose I simulate new data with my model parameters that I infer, do I see similar discrepancies under those simulations? So it's kind of like it's in line with parametric bootstrap, if you will. And then you try to compute this distribution and compare to this distribution, or you don't really have to compare. You can also calculate the probability of this event, or you can look actually at those distributions and see how different they are. So here the probability is taking over the joint space of your predicted data and your model parameters, which is factored into predicted data given your model parameters, and your model parameters come from the from the posterior distribution of your data and your null hypothesis. It's a little convoluted, but just think about the following experiment. You have we did inference, we estimated parameters, we get our discrepancy measure, just one value, and then you try to simulate data under this inferred parameter, and you see that no matter how hard you simulate, your distribution is shifted from what you observe. Your distribution here and your number that you get in the first place is way on the left, on the right tail. So that's the basic idea. So in practice, how we actually achieve sampling from this posterior distribution? Well, in the MCMC iteration, for each iteration in your MCMC, you simply generate a new replicate, dataset replicate, and you just define those indicator functions which allow you to compute those probabilities. So in practice, it's simple to implement as long as you have an MCMC sample that can do the simulation from your posterior. So this is an example, sort of a primate evolution example. We have two traits. So this is just a sample of a tree. Now I'll make it a little more readable, like this. So here I discard branch length, so you can see what the traits are. So they're all aligned. So those are primate species, and what we observe here is two traits: estrus advertisement and mating system. And there's a hypothesis among evolutionary biologists that those traits should correlate. So when you have multi-mating systems, females should advertise their estrus, and therefore the traits are correlated. So 60 old world monkeys and great apes, this phylogeny. Now, what I did, I actually took a sample of phylogenies and a sample of parameters that govern the evolution of those two traits. And the hypothesis is they're independent. Before there was some evidence was obtained by Pagel that indeed there is some correlation that you can see in the data. And notice it's the same situation as Jonathan described for the HIV. And we have two traits, and if you don't account for common history, it's not surprising that those two traits are always zero here, because they didn't have time to evolve to anything else. So the common ancestor, for example, of this clade, clearly all of macaques inherited both once from their common ancestor. So you cannot really assume that it's all independent and just count things and do the standard test of independence. You should account for this correlation among macaques, for example. So if you do these Bayesian p-values, so that's discrepancy measure we use. So basically we consider all possible pairs and calculate observed versus expected, and their discrepancy. Now what you see, it's observed versus predicted. This is the joint plot, because those are marginals, and marginals do not tell you everything. And the p-value is sort of small. So I should warn you. I will not talk too much about the dangers of doing this Bayesian analysis. The problem, it's a good method for just looking at the data and seeing how well it agrees with your model. So it's a good sort of just an exploratory tool. Don't try to do any scanning with it. I beg you, because people are doing scanning with these methods, and this is just plain wrong. The null distribution of this p-value is not uniform. It's usually, we don't really know what it is. Usually it's concentrated around 1/2 and sort of decays quickly, but it's not uniform. So you cannot do when you do scanning, you really expect that you will not infer something 5% of the time or something. So you cannot make these statements when you have those p-values. Moreover, those p-values change depending on the discrepancy measure you use. The distribution changes. So if you cannot really even compare two discrepancy measures, you have to calibrate them first. And there is a method for doing that, but it's computationally expensive. Unfortunately, I don't think there's a realization in the field that you cannot do the scanning with those things. So that's just an illustration. If you had, so here I simulated data under independence just to show you how a good model, or if our null was appropriate, how things would look like. So of course, your predicted is more dispersed than you observed, but they overlap a lot, and you don't have any discrepancies. Your p-value is very high, so it's around 50%, I guess. So it is concentrated. Actually, we know that the mean is, we know that the mode is at 50% under the null. So this is another fun example. This is cultural evolution example. I particularly enjoyed because I spotted a mistake in the data here, which happens rarely to me. So again, this is the real tree. I just collapsed now. I ignore the branch length just for the purposes of illustration of the traits and the names. So those are 51 Indo-European languages, and now traits are cultural. So we treat culture. So if we go to cultural evolution now, DNA becomes language data, and evolutionary traits or morphological traits become cultural traits. So it's a controversial topic whether you can do it or not, and when it's appropriate and when it's not. But nevertheless, computational linguistics evolves hand in hand with phylogenetics. We use the same tools. So again, I estimated had a sample of phylogenies, and they were estimated on some cognate classes. You think about them as just your genetic information. And hypothesis here, so we have two traits, wealth transfer and marriage system. And supposedly, a presence or absence of dowry versus bridewealth is correlated with monogamy and polygyny. And there's some explanations why it could be so. So the mistake I spotted was that Ukraine ended up having polygyny, and I'm Ukrainian, so I know that it's not true. So I corrected that mistake, but it turns out the authors, I guess, noticed that too, and they corrected it afterwards. But in the publication that I looked up, it still wasn't correct. So when we do the analysis, again, we have I didn't do that. We found a strong support for correlated evolution, and again, you see a distribution does not look like your distribution. It looks like you have those sort of very, very strange discrepancy measures that could not come from your predicted distributions if your null model was correct. Now, so this is some exotic applications of those discrepancy measures, but you can envision that those applications can be moved to this HIV, and I will certainly try moving those applications in those directions. But there are many others. I didn't talk about counting process at all, like how you count mutations. So just briefly, I just want to give you a flavor of what kind of things you can do with counting processes and sort of mapping mutations on trees. For example, you can take an HIV tree. So this is intra-host HIV evolution, about 100 sequences, sampled across 10 years. Notice the structure of the tree is very characteristic of this type of analysis. When you have serially sampled data of HIV or influenza, you get a sort of backbone, and you have those lineages that die out quickly. So you have this structure very sort of specific for those type of design when you sample sequences across time, and the sampling times, the differences between sampling times is actually significant enough to make difference in phylogenetic estimation. So this is just one snapshot. Of course, we get a whole collection, a cloud of trees. Now, what you can do, you can using a DNA model, for example, you can ask questions. Well, what's the expected number of synonymous and versus non-synonymous mutations? And synonymous mutations are those mutations that do not cause change on amino acid level. So you change your nucleotide, but your amino acid doesn't change. And the non-synonymous is the one that changes amino acid. So non-synonymous mutations are, they do change the supposedly fitness of the protein, although in HIV it's a controversial topic. You cannot really claim that synonymous mutations are neutral because you do change perhaps RNA structure of HIV. So, but for the moment we will ignore that. So for each branch, we can calculate this expected number of synonymous and non-synonymous mutations that we have. And then, for example, you can group branches and ask, well, if I group terminal versus internal branches, what's the fraction of non-synonymous mutations that I see? And under your model that you hope is true, there's no difference between external and internal branches. So those two distributions should overlap, and they should actually be together, have the same mean, and possibly different variance simply because of the sampling. But you see that they're very different, well, it's nothing new. I mean, we suspect that some branches have different rates of evolution, and especially non-synonymous mutation. There's several biological stories you can make up, but we don't. The true answer is we don't really know what's going on. We're just trying to explore. But you can envision counting mutations differently. You don't have to count non-synonymous and synonymous mutations. You can label those mutations according to what kind of amino acids they produce, hydrophobic, hydrophilic, if you're acting on amino acid space. Or you can sort of make up those labeling sets according to what kind of volatility your codons have. So some codons are more prone to having non-synonymous mutations sort of occurring from them, and some are surrounded mostly by synonymous codons. So you can envision creating all sorts of counting measures which can bring us a little closer to actually understanding how this beast evolves. Because no matter how hard we study, it doesn't seem like we understand anything at all about HIV evolution. And I will conclude just by giving you some references. So Rasmus Nielsen came up with this original paper of using simulations. No matter how inefficient they are, it was and still a breakthrough because we cannot still convince too many people to use this methodology. Most of the time you see people use sophisticated Bayesian tools. They reconstruct, they fit complicated models. They're fairly savvy at model selection. But then at the end they say, well, now we have a tree, and we use parsimony to reconstruct the history. And it's like, well, why did you do all of that? Why did you do all this Bayesian approach if you, at the end, you just gave it up, and you just gave some strange point estimate of evolutionary history instead of characterizing the whole distribution that you have? So I still we have a lot of work to do to convince practitioners to use this stochastic mapping. And we hope that our results make it a little easier, and we will produce some useful software that will allow to do that. This is the paper I mentioned by John Halzenbeck, Rasmus Nielsen, John Balbeck on using stochastic mapping for doing this chi-squared, kind of Bayesian chi-squared test for independence with the posterior predictive p-values. So this is some Boll and Milne. They have nice results for counting processes. So I've learned a lot about from those guys on how to compute things on one branch. This is a nice book that has I like especially the name, the algorithmic probability. I think it's what this flavor of probability that was used in statistical genetics a lot. When you approach a problem, you don't really care about about limits. You care about how do I compute probability in practice? How do I get a number back? How do I get my likelihood? How do I get my posterior probability? So in this paper, we summarized results. So this is one branch calculations that I described. I omitted this in this talk, but it goes into details how to efficiently compute things on one branch. And this is the Pagel paper that I alluded to when I showed you the primate evolution example. And so this Mark Suchard was my advisor at UCLA. Todd Oakley originally motivated this problem by posing for asking us how to test the independent evolution hypothesis, which we also can do with posterior mapping without any simulations. I didn't have time to talk about that. So he was he motivated us to actually look at the problem and then realize that it has an easy solution and solve it. So we're very grateful to him. Andrew emailed me some data that I showed you, and Laura Fortunata gave me this language data to play with. and thank you for your attention.